I'd like to thank the organizers for inviting me to be part of this great meeting and allowing me to share some of our work on gene and stem cell therapy for inherited retinal diseases. I'm pleased to say that I have no personal financial interest in any of the work that I'll talk about today. My objectives are, first, to provide you with the classification system that I use to organize and think about all the different inherited eye diseases that a retina doctor is likely to encounter in his or her practice. Next, I'll give you a sense of the magnitude of the inherited retinal disease problem, and for the majority of the talk, I will outline the strategy that we're pursuing to devise affordable gene and stem cell-based treatments for these diseases. The most powerful use of a classification system like the one I've given you in the handout is to focus the efforts of a genetic testing laboratory on a relatively small number of genes in each patient. There's so much noise in the human genome that unless you have a pretty crisp clinical diagnosis before genetic testing, you will have an unacceptably high risk of making a mistake in your molecular diagnosis. I recently reviewed the genetic testing results from 1,000 consecutive families seen in my clinic at the University of Iowa, and we were able to identify disease-causing mutations in 760 of these families for an average cost of $980 per family. These mutations were distributed across 104 different genes. The most common gene was ABCA4, the gene that causes Stargardt disease, which was present in 174 of the 1,000 families. But 91 of the 104 genes caused disease in less than 1% of the cohort. More than 75% of the genes are small enough to fit into an adeno-associated virus, which has been shown to be a safe and effective gene therapy vector in a number of human diseases, including inherited retinal disease. The task before our field now is to test every family affected with an inherited retinal disease to identify the causative gene and to provide disease-arresting gene therapy to every patient with early disease and vision-restoring retinal cell transplantation to every patient with late disease. To accomplish this, we need to keep the costs low. Genetic tests for less than $1,000, gene therapy for less than $20,000, and stem cell transplantation for less than $50,000. This is because without some type of very significant governmental or actuarial wealth redistribution, which doesn't seem very likely in the foreseeable future, the vast majority of people in the U.S. will not be able to afford treatments at the $1 million price point that many commercial entities are proposing. I think that it will be feasible to develop inexpensive, effective gene-based treatments for dozens of these diseases, and patient-derived stem cells will play a major role in this outcome. In a number of cases, we'll be able to elucidate the pathophysiologic mechanism and demonstrate the correction of the disease phenotype using human cells instead of animal models. Here's what our current pipeline looks like. We obtain a blood sample and skin biopsy from every new patient with inherited retinal disease, and we use the DNA to identify the family's disease-causing mutations and the skin biopsy to establish a cell line. In a subset of the individuals, the cells are reprogrammed into induced pluripotent stem cells and differentiated into the cell types that are most relevant for the disease gene in the family. These cells are then used to investigate the cellular phenotype associated with the mutation and to investigate the efficacy of drug and gene-based therapeutics. If you own your own GMP facility, you can manufacture a batch of therapeutic AAV vector capable of treating over 100 patients for less than $5,000 per patient. We built the DESI facility at the University of Iowa to manufacture viral vectors and transplantable retinal cells in a completely nonprofit fashion. And if anyone is interested in building a similar facility, our blueprints, instrument lists, protocols, and standard operating procedures are all freely available. The University of Iowa hospitals have also agreed to let us perform the subretinal injection surgeries at their cost, and as a result, a phase 1-2 clinical trial of gene therapy is projected to cost less than $20,000 per patient to conduct. However, if we want to cover the entire 104 gene landscape with clinical trials soon enough to be meaningful to any of the patients we're seeing in the clinic today, we'll need to initiate several new clinical trials per year. We think such trials will be feasible using little more than philanthropic dollars by packaging the vectors in a nonprofit GMP facility and reusing the FDA IND documents from the initial studies for the later ones. So what about patients like this 62-year-old man with advanced retinitis pigmentosa who have just a few degrees of vision left? Although he has some relatively preserved retina in the center of his macula, he has very extensive degeneration of the outer retina and retinal pigment epithelium everywhere else. As you know, there are really just 3 neurons required to detect photons in the retina and send action potentials down the optic nerve to the brain: photoreceptors, bipolar cells, and ganglion cells. The photoreceptors are lost in retinitis pigmentosa, but the bipolar cells and ganglion cells can persist for decades, even after a patient has progressed to no light perception. This raises the possibility of restoring vision by transplanting stem cell-derived photoreceptor precursors into the subretinal space of these patients. Indeed, this has already been accomplished by a number of groups using various animal models of photoreceptor degeneration. In this experiment, my colleague Bud Tucker injected iPSC-derived photoreceptor precursor cells into blind mice and observed increasing electroretinographic activity with increasing doses of the injected cells. As we translate this idea into humans, we think it will be important to derive the transplanted cells from the patients for whom they are intended. There's a growing body of evidence that retinal allografts are associated with a robust immune response, even when the recipient has a normal retina prior to the transplant. And although it might be possible to prevent this with lifelong immunosuppression, the drugs needed to do this are expensive and are quite toxic to other major organs in the body. If new retinal cells can be derived from a patient's own tissue, they will be the best possible immunologic match and require the least immunomodulation. To make patient-derived photoreceptor precursor cells, we first take a small skin biopsy into the GMP facility and use it to establish a fibroblast culture. These cells are transiently treated with 4 pluripotency factors that essentially erase the cell's differentiation state and turn them into induced pluripotent stem cells. These iPS cells are then cultured with a series of additional growth factors chosen to drive forebrain development, and after about 30 days, one can observe eye cup-like structures extending from the presumptive brain tissue. These eye cups can be surgically removed and cultured with additional growth factors chosen to foster retinal development. At the time of dissection, the eye cups have a lot of proliferating cells, as shown by the marker Ki67. 15 days later, the proliferation slows down, and the retinal markers SOX2 and VISX2 appear. By day 60, the photoreceptor marker OTX2 appears in some rosettes, and by day 90, recoverin and CRX appear. At about 120 days, cells with intense recoverin expression are present, and it is these photoreceptor precursor cells that we think are optimal for therapeutic transplantation. Although one could simply dissociate these cells and inject them as a bolus under the retina, we know from extensive animal studies that very few of the cells would survive and integrate if we transplanted them in this fashion. So we've been using 2-photon 3D lithography to create laminin-coated biopolymer scaffolds. Such scaffolds increase the survival and integration of transplanted cells more than 50-fold in animal models. Here's some scanning EMs of scaffolds with holes that are 10 microns in diameter, just large enough to admit the photoreceptor precursor cells. Here you see a series of confocal images of a loaded scaffold showing very uniform loading. There are small perpendicular channels connecting each of the wells in 2 dimensions that will allow contact among the neurons as they mature within the graft. For our first human trials, we're planning to transplant a 4.8 millimeter circular graft containing approximately 500,000 photoreceptor precursors into completely blind patients. By placing the graft in the macula, the transplanted cells will have the greatest chance of synapsing with viable bipolar cells or ganglion cells. However, just like gene therapy, all this progress won't meaningfully impact heritable blindness until we can deliver it on a whole population scale at a cost that society can afford. This will undoubtedly require the use of automation. These little movie clips show a couple of the robots that we are currently adapting to perform the thousands of repetitive tasks that will be required to manufacture patient-derived, polymer-supported retinal grafts at a scale that will make the surgeons rate limiting instead of the grafts. So let me leave you with 3 goals for our field to work together to achieve. Genetic testing for everyone who needs it for less than $1,000 per patient, gene therapy for less than $20,000 per patient, and stem cell transplantation for less than $50,000 per patient. I'd like to thank all the faculty and staff of the Wynn Institute for Vision Research who contributed to this work, and especially Bud Tucker, who's the creator and director of the DESI Stem Cell Facility.